As she said, my name's Lindsay Frankart. I'm from DePuy Synthes, and I am a toxicologist there. So today I'm going to talk to you about non-volatile residue reconciliation, so like the expectations regulators have and reality. It's going to be a little different than a lot of the presentations you've seen today. I'm not going to tell you like what to do according to the standards. I'm going to tell you a story. So this is a story of non-volatile residue reconciliation in a recent FDA submission from the perspective of a toxicologist. So I am not a chemist at all. By training, I'm a physiologist. Toxicologist is my job, and this was a fun experience we had with the FDA. So I'm kind of skipping over a lot of the device details just to kind of keep it anonymous, but I work for DePuy Synthes. It's an orthopedic company, part of Johnson & Johnson. So as you can imagine, it's an orthopedic device, long-term implantable, skeletally mature patients, and the surgeons could use 1 to 2 depending on their needs. It's as simple as a medical device comes. So single, solid, metallic device, conformed to an ASTM, and was passivated with nitric acid as its final manufacturing process. So about as simple as you get for a medical device. We sent it for chemical characterization according to Part 18, which I'm not going to cover because we've talked about that in depth. It was sent to a US CRO laboratory. I'll just call them Lab A for anonymity, in October of 2020. The results were available May of 2021, and I joined DePuy Synthes March of 2021. So I was not there when the study was placed, but was there for the results at the end. And the team had a 510K submission planned, all scheduled, you know how project teams do that, for July of 2021. So regardless, we were submitting in July, and they gave me a couple months to get the TRA done and everything ready. So we did exhaustive extraction, pretty simple, basic, straightforward. The test articles weren't cut in any way. They were extracted in glass containers, and the NVR measurements were taken through gravimetric analysis. You can see the volume of vehicle was a little variable depending on the size of the container they used. They couldn't follow the Part 12 standard because they couldn't get the device, imagine a large metallic device, to fit in something simple that they could get that right ratio. So they used a large volume and then concentrated it down. And here's our results. I'm guessing a lot of you in the room can see that it's a little odd or unexpected. We did not get the results until the study was complete. So nobody told us anything unexpected happened. We got the results in May, and this was it. So the key to look at is that purified water. It's a little all over the place. There's no pattern. It was low, it was high, it was low again, it was high again. And we came out with 3.9 milligrams in the purified water. Isopropanol and hexane had pretty low amounts, kind of as expected, so we didn't really worry about it. The team came to me and said, remember, we have our 510K submission for July. What are you going to do? I was like, well, the levels of NVR are concerning. Like, why would we have that? There's no pattern. It doesn't really make sense, but it's just NVR. Like, they dried it down, they weighed it, and it's only used for, analytical to determine the exhaustive extraction conditions. So we looked at the analytical data that was there, and that data was sound. It looked good, it made sense. The levels were as expected. So we evaluated the toxicity, created a TRA, and we did not do biological testing on this device because we already had both in vivo and in vitro biological testing on a sister device that had very similar manufacturing processes and the exact same materials of construction. So they were so close, we decided not to repeat that biological testing, leverage that, and do this TRA. So we submitted in July. The project team, high fives all around, yay, we met our deadline. And I was like, hold on, guys, it's the FDA. They're gonna have some questions. And we did. Our first questions came back in September. And the major one was on NVR. So I don't expect you to read this, okay? I wanted to show you guys what their question was because it's never just a question, it's, a story. The keys are, there was a large mass of NVR, that 3.9 milligrams in the purified water extract. Why did you have such a high amount in the NVR and so little in the analytical data? We're concerned that you didn't evaluate all of the compounds for toxicity. Please compare that NVR to what you found in your analytical data and make sure that it won't impact your toxicological conclusions. So we got the project team together and we looked at it. We kind of investigated what was going on here. So the difference is here on the screen. You can see for the purified water, we had 3.9 milligrams of NVR and 0.095 milligrams detected in the analytical extracts. That's what the FDA was concerned about, that 3.8 milligram difference that we couldn't explain with the analytical data. The isopropanol and the hexane behaved kind of as expected. There were some differences, but not enough for the FDA to be concerned. So we contacted our lab. I said, hey guys, what happened with this NVR? Anything odd? Help us explain this to the FDA. They're asking this question. And this was really our major biocomp question. So once we got past this, we'd be all set. The lab helped us write a response. And they're like, oh, it's probably high molecular weight compounds. Those can get detected by the NVR because they have weight, but they're not detected by the analytical testing. So it's nothing to worry about. They don't have a toxicological concern. We can just write this up and we'll give it to the FDA, and we'll also tell them some stuff about how, there's no analytical technique that can capture everything. Chemical characterization, is not perfect. So we're like, perfect. We'll send that to the FDA. We sent it to them in November. They came back in questions in December. Their questions, again, it was the whole story. Don't expect you to read that. But really it was like, hey, you said your NVR includes high molecular weight compounds, but you didn't prove it. So go back and prove it, was kind of what they said, and have fun proving it, show us that this is here. They really wanted to know, is there a risk? That stuff that you measured in the NVR, what is it? And is it a risk to patients? So one of the lines that caught our attention was the last line there that said, demonstrate that this does not pose a biological risk to the final finished device. So the team got together and said, okay, we don't know how to explain the NVR. We can do testing. What do we do? How do we mitigate this so that we're not continually having this conversation with the FDA over and over again? So the team decided to do biological testing. That last line said, show that it doesn't have a biological risk. We had that testing from the sister device that we used. So we went ahead and we just did testing on this device. We didn't do all the testing. The team did not want to do the long-term animal studies. So we did basically all of the shorter-term animal studies and in vitro studies that we could. We looked at the manufacturing aids. So I asked all of our suppliers for the SDS sheets. Had to clarify that I needed them in English, because that's always fun when you get them not in English, if you can't read another language. So we got them in English, and we looked at them for any health hazard statements regarding genotoxicity, systemic toxicity, carcinogenicity, to kind of evaluate if there was any risk to any manufacturing aids that were left on the device. We included that in the TRA, and we did gel permeation chromatography to find those high molecular weight species that we told the FDA was probably the cause. So we sent 4 devices to Lab A again, and 2 of them were extracted in purified water using the same timeline that the analytical testing was, and 2 in hexane for the same timeline as the analytical testing. And we created 2 samples of each extract, and we found nothing in the purified water, absolutely nothing. But we did find a peak in each of the hexane extracts. So there were some high molecular weight compounds there, just not where we expected to find them. So we're like, okay, well, we told the FDA it was probably high molecular weight compounds, but we've got all this biological testing now, and we looked at the manufacturing aids. It'll be fine. So we told them, here's all the biological testing, here's our GPC testing, and that new updated TRA that included the manufacturing results. So we told them, using a weight of evidence approach, everything's fine. Like, the device is safe for patients. Pay no attention to that NVR. It's obviously kind of, like an artifact from the lab. The device is fine. We sent them the response in March, and they came back in April with questions. And as you can imagine, same question. What is that 3.8 milligrams? And at this point, we're like, we don't know what that 3.8 milligrams is. This is why we're doing all this other stuff. So their key at this point was, if we couldn't prove that 3.8 milligrams, what it was, they didn't trust our analytical data or our TRA. So all of the work we had done, all the analytical data, really, how could they trust it if we had this discrepancy? So they gave us kind of 4 things to do. Provide a justification that explains what the NVR data is. Okay, well, we can't do that yet. Show that the analytical data supports a tox risk assessment. Struggling with that one too because of that first one. Look at all the manufacturing residuals. So we did that with those SDS sheets. We put together a nice section in the TRA. And then if we can't mitigate the risk, we might have to do those long-term animal studies that we were trying to avoid by doing the chemical characterization data in the first place. So as you can imagine, the project team's already looking at us going, we submitted in July and now it's April. Like, 1, we're running out of time with the FDA. And 2, we're not gonna wait to do long-term systemic toxicity studies. Like, what are you thinking? So we did have quite a few calls with the FDA at this time. A lot of discussions, a lot of questions, and we got them to agree to allowing us to do more NVR testing. It was not what they asked for, but it was the fastest thing we could think of to get us through this conversation of what is at 3.8 milligrams. So we did more NVR testing with Lab A, because they were our laboratory partner for this project. We wanted to see if it was an artifact of that one device. We sent them 3 lots with either 2 or 3 samples per lot. And for a lot of them, we found no NVR, no measurable NVR. For one of them, second one from the bottom, and at the bottom row now is our original data. So one was very similar to our original data at 4.2 milligrams. And our original data was 3.9. We had a couple of them from one lot that had 1.0. So we all look at each other and get this back, and we're like, we can't send this to the FDA. And you can imagine that conversation. We're like, we just now proved that there's lot to lie and inter-lot variability. Like, how do we send this to the FDA? So we called them up and we're like, hey, you know how we said we were gonna do more NVR testing? We're gonna do more NVR testing. And they kind of looked at us like we were stupid, but they're like, fine, just go ahead. So we sent devices to a different US-based laboratory, and we sent 5 lots this time, each with 2 devices. And they found nothing, which is pretty much what we had expected originally, because it's a solid metallic device being extracted in purified water. We even had them keep going out the full time that Lab A did to make sure that we didn't have anything pop up at the end unexpectedly, but they found nothing. We even sent them lots that we had tested at Lab A, and there was variability between the 2 labs for those lots. So that one that was 4.2 milligrams that we couldn't explain, same lot was tested at Lab B, and they found nothing. So this was good news, finally. The project team was quite relieved that this was the result we got from Lab B. And then they were kind of like, well, why didn't we use Lab B in the first place? That's a whole nother conversation. But we were able to put together a response for the FDA. And we were able to say that the results from the NVR testing were artifacts. They weren't indicative of what would come off of a final finished device in a patient. They weren't even indicative of what would come off a final finished device at another laboratory. So, the device itself, when we looked at that other laboratory data, was safe. We concluded that the analytical chemistry was sound, and it was analyzed for a toxicological risk, showed there were no concerns toxicologically. We looked at biocompatibility testing on the device. We looked at the manufacturing aids for the device, and everything was safe and that there were no concerns for long-term or systemic effects. We sent that to the FDA in August, and to everybody's kind of surprise and pleasure, we got our 510(k) clearance in September. So why am I telling you all this? One, it's kind of a funny story that I spent like over a year of my life dealing with. But two, there's a lot of things to learn from it. So one, before this specific project, I looked at NVR and said, oh, it's just NVR. Like, the lab weighed it, they measured it, and they said, okay, this is how much we have to do to extract to get the amount we need to do for exhaustive extraction. Like, this is just our exhaustive endpoint. I, as a toxicologist for years, had looked at it and said, okay, yep, they met exhaustive at this, they extracted it for that, and never looked at the NVR data again. This project made me realize NVR is important. If you are submitting to the FDA, I know we're in Europe, so I don't know how many of you submit to the FDA, but if you do, be prepared for the FDA to question this. Since this project started and we submitted, we received another question exactly like this on our other 510(k) submission. Totally different device, totally different circumstances. The original question was almost word for word. Explain the difference between your MVR and your analytical results. So pay attention to it. Your laboratory partner is important. That's probably the biggest thing we saw with this is, the difference between lab A and lab B. Pick your lab partner like you're picking like an actual date. It's important. So you need to pick a good lab partner and you need communication. The fact that we didn't hear anything about the MVR results until the final study was submitted was kind of a struggle. Had we heard about that in January, we could have done something ahead of time to mitigate that. So my study director for a current project here at Nelson, I'll probably run into him today, I'm asking all the time, are those MVR results ready? What was it? What are we doing with those? But I want to know. This is important to me now, and it's something I stay on. I can't really look at the analytical data when it comes off, but I can definitely ask about those NVR results. And if possible, I'm asking now to, have something documented. Have the lab look at it, do something to mitigate that so I don't have to spend a whole year trying to figure out what that NVR was. Some labs are moving to chromatographic techniques for NVR instead of just the gravimetric, or for exhaustiveness. That's always an option. The FDA is a little iffy on how they like that, but that's an option. And then if you find yourself in a spot like we did where we were kind of stuck, and the FDA basically said, prove that this, tell us what this is or do more testing. Doing lot-to-lot variation between an NVR might be a good way to get out of it. If you ever find yourself in kind of this situation, that's always an option. So I hope that you learned a little bit from my story and had a kind of a laugh at our expense. But it was very interesting and kind of a nice thing to learn. So thank you.